Good day everyone. Today I'll be presenting a study on the bacterial composition of the urinary microbiome in South African men with and without Chlamydia trachomatis infection. My name is Kehinde C Mofolorunsho from the School of Clinical Medicine, University of KwaZulu-Natal. The human microbiome, consisting of 38 trillion commensal and pathogenic microorganisms, play a crucial role in maintaining health and regulating physiological homeostasis, energy metabolism, and immune-related bowel processes. However, changes in the microbiome can lead to pathological conditions like allergies, inflammatory bowel disease, and bacterial vaginosis. The urinary tract, initially thought to be sterile, has been reported over the last decade to harbor unique bacterial communities, with alterations in its composition implicated in varying urinary tract symptoms and disorders. The urinary microbiome of males, especially those of men who have sex with men, is poorly described. However, it has been proposed that the bacterial colonization of the male urethra may influence the risk of sexually transmitted infections, including Chlamydia trachomatis. Men who have sex with men are on the studied population at high risk for genitourinary diseases, and exposure to the gut and oral microbiome through unprotected sexual behaviors may lead to shifts in the urinary microbiome of men who have sex with men that increase preference for genitourinary diseases. This study aimed to assess the bacterial composition of the urethral microbiome in a cohort of men who have sex with men with and without Chlamydia trachomatis. This study used urine samples from 200 sexually active MSM aged 18 and older attending the King Edward Hospital and the Aurum Institute in Durban, South Africa. A total of 10 ml of each urine sample was centrifuged, the supernatant discarded, and the recovered pellets subjected to further molecular analysis. The samples were processed and analyzed using the PureLink microbiome kit and nanodrop spectrophotometer. C. trachomatis was detected using applied biosystems Tachman assays. The 16S ribosomal nucleic acid gene was amplified using the following forward primer and reverse primer. DNA library was prepared using the PCR products following the Illumina nano DNA sample preparation protocol. The 16S rRNA gene sequences were analyzed using the Quantitative Insights into Microbial Ecology 2 pipeline. Taxonomy for the 16S rRNA reads were assigned using the Silver 132 database. The taxonomic data was further analyzed using RStudio, an integrated development environment for the programming language R. Principal coordinate analysis and permutational analysis of variants were used to visualize taxonomic structures of microbial communities and assess beta diversity differences between sampled groups. Results. This study involving 200 MSM tested for C. trachomatis infection found that 6% of the samples tested positive. To maintain a balanced sample count, 24 samples were sequenced. One positive sample was excluded due to insufficient DNA, resulting in 23 samples. High throughput sequencing yielded between 291 and 249,759 reads. Here is a Venn diagram showing the number of unique and shared amplicon sequence variants between samples from C. trachomatis infected group, the one in blue, and uninfected group in green. A total of 455 amplicon sequence variants were identified across all samples, with 92 unique to positive samples, 302 unique to negative samples, and 61 shared between both. Sample diversity at phylum level classified most weeds as Firmicutes, Actinobacteria, Proteobacteria, Bacteroidetes, and Physiobacteria. Differences in the distribution of these phyla among samples were also observed. Firmicutes were present in 22 of the urine samples, while Actinobacteria and Proteobacteria were identified in 15 and 11 urine samples respectively. Bacteroidetes and Physiobacteria were present in 7 and 6 urine samples. However, Verrucomicrobia were detected in only 2 urine samples. At genus level, a diverse consortium in all samples with dominant genera including Streptococcus, Corynebacterium, Staphylococcus, Prevotella, Gardnerella, and Lactobacillus were identified. Alpha diversity metrics showed a slight increase in microbial diversity in positive samples, but this was not significant. Diversity measures of microbial species recovered from urine samples were not different between C. trachomatis positive and C. trachomatis negative groups. Principal coordinates analysis showed that the microbiome of C. trachomatis infected MSM was not clearly separated from uninfected samples. The gray dots indicate urine samples from MSM positive for C. trachomatis, and the red dots correspond to urine samples from MSM negative for C. trachomatis. The percent variations explained by each principal coordinate are shown in parentheses. C. trachomatis, a common sexually transmitted infection, is a significant public health concern globally, with 131 million people infected annually. To the best of our knowledge, there is a dearth of information in South Africa on the bacterial microbiota in urine from MSM with and without C. trachomatis. In this study, the urine microbiome in the MSM with and without C. trachomatis were investigated. Our study revealed a high diversity as study participants harbored diverse taxons. This is in consonance with findings from a previous study which reported that a large diversity of microbial species were present in the urinary microbiota of MSM. The urinary microbiota composition at the genus level showed a higher abundance of Streptococcus, Corynebacterium, and Staphylococcus in both the C. trachomatis uninfected and infected groups. This was consistent with other findings where similar organisms in a cohort of asymptomatic MSM were also recovered. This study found that Lactobacillus and Prevotella were found in the urethral microbiota of MSM. Other genera were Schnitzia and Gardnerella. In a prior study involving males visiting an STI clinic, Lactobacillus and Streptococcus were among the most frequently detected genera. That same study also showed that STIs were associated with organisms not usually related to the male urinary tract, such as Schnitzia and Prevotella. Urotypes such as those dominated by Prevotella and Lactobacillus in the female urinary microbiota have been described as members of the male urinary microbiota. Changes in the urinary microbiota of MSM may result from exposure to unique microenvironments such as the gut and oral microbiota through anal and oral sex. In the present study, we detected Prevotella from urine samples in both MSM with and without C. trachomatis. This finding is in agreement with a study which also found that Prevotella was one of the most frequently recovered genera from urine samples in MSM. This finding also suggested a potential gut reservoir as the source of Prevotella in the urinary microbiota, with the recovery of Prevotella in 59% of MSM who reported recent insertive anal sex. Our study also identified Schnitzia, a genus commonly observed in the vaginal microbial communities of women with bacterial vaginosis. Similar with our finding, this genus was among the microorganisms recovered in the study involving cohort of healthy MSM. A limitation of this study is that DNA libraries generated for the target metagenomics for this study did not generate adequate sequencing data due to low number of sequences. Also, the sample size used in this study was small. In conclusion, this study adds to the growing literature highlighting the urinary microbiome in MSM. It is clear from our study that unique microbial communities, especially those associated with the female urinary tract, are present in MSM. Therefore, continued research into the role of the urinary microbiota in urinary health of this understudied population is required. This will provide clarity as to whether these microorganisms impact risk for sexually transmitted infections. These are some of the references used in this study. Thank you for listening.